Hi, my name is Josh Wolf. My project is entitled Electrically Stimulated Lipid Secretion of Botracoccus brownie. My project aims at using electricity as a means to efficiently extract oil from algae to be applied to alternative energies. In my project, I used an electrical stimulation technique similar to that used in neurological studies. My project uses a very low voltage, low amperage, and low frequency pulsation to initiate latent functions of exocytosis in the algae. The goal of my research was to lower the economic strain required to extract oil from algae. The application of this process has very real-world applications because it can be used industrially to produce oil from algae and make it economically competitive with crude oil. This system can also be used in developing nations as a means of easy acquisition of alternative energy. My project started in 9th grade when I started researching algae as a solution to environmental problems. This interest sparked from me taking trips with my grandparents to Itasca State Park, where I got to see all these amazing, huge trees, and it really got me thinking about what the role biology has in the environment. I started thinking more about how if Itasca was relocated to an area that produces a high level of CO2 like the Twin Cities, how those trees could benefit the atmosphere by absorbing that CO2. So effectively, I started researching how the power of those humongous trees in Itasca State Park could be brought to the cities to take up that CO2. That's how I got to algae. Algae has been known to have CO2 capture rates and oil production rates thousands and thousands of times higher than any other plant. I began growing cultures of algae in my room in just water bottles. This passion of mine to study how this organism grew came from just water bottles to making larger tanks to seeing really how nutrient concentrations can affect the growth rate of algae and how aeration can affect the growth of algae, and all of these small little things that began to compose in themselves into a complicated method of scientific research. When I started talking with my AP biology teacher about my somewhat weird interest with studying algae, he was able to instruct me on how proper scientific research is conducted. Things like making sure your samples are sterile, there's no contamination from outside organisms, the little things that you wouldn't necessarily think about without having formal training. So this interest of mine started to grow from just studying little bottles of algae all the way until I started to have my first eureka moment with algae when my AP Bio teacher was talking about the function of neurons and how they're constantly releasing neurotransmitters into the synaptic cleft. And I thought, you know, these neurotransmitters are contained in bodies called vesicles. And that's exactly what they call it, vesicles that hold oil inside of algae. So I thought, all of these companies in California, New Mexico, Nevada are having such a hard time economically removing this oil from algae that are inside of these vesicles, whereas neurons get rid of stuff that's inside of their vesicles billions and billions of times a second in the human brain. So I wanted to try and figure out a correlation that happens between algae and neurons that would allow algae to have the same release of lipids that neurons do with neurotransmitters. I took this concept and started developing a model that would allow for me to change variables like voltage and amperage and frequency into a culture of algae, and how that would affect or not affect the production or secretion rather of oil from algae. And this was kind of, as I continued to do this research of something I thought would make sense as an efficient process to remove lipids from algae, I had to develop a stronger skill set for research analysis, data analysis, even the things that I thought would be not a big deal, like setting up a lab report or constructing the lab itself. All of these were traits that I had to get really strong before I could make any sort of impact. Otherwise, my data could be insignificant, or I could have had a flaw somewhere in my experimentation, and that would completely compromise the reliability of my data. So these were things that I had to get really strong before I could continue on. And this was what my biology teacher had helped me with. And by the end of him helping me out, I finally decided that I was going to enter my research into the science fair and just see what happens. So I entered in the St. Cloud State Regional Science Fair, where I got third place, oddly enough, for my research. But that was my first opportunity where I could see like-minded students in academia and how competitive intellectually that environment is. Then from that regional competition, I was sent on to the state competition, where I had some time to formalize some of my vocabulary, some of my data analysis skills, and just little things that I picked up along the way from my encounters with peers at the regional science fair. And then I went to compete at the state science fair. At the state science fair, I ended up taking first place, and I received the 3M Innovation Award for the most innovative project that had a very high applicability to either industry or medicine. So this was kind of the turning moment of, I have this odd interest in algae as a means for biofuel production, to actually becoming an influential figure in the field of alternative energies, because I had presented my research in front of doctoral students, in front of doctors at the state science fair, and they all had given me this great appraisal for my research and its application. So that approval that I had gotten from them kind of propelled me from this naive sort of research that I had been doing to saying, you know, this is a pretty serious life opportunity for me to have, and this is something that I can do for my future. Following my success at the state science fair, one of my teachers started to help me move from my naivety of research to actually real-world application by helping me get set up with a patent attorney, where I'm now patenting my my research on algae lipid secretion methods, because it's now one of the most efficient methods currently published for extracting oil from algae. I hope to take this patent and apply it and license the technology out to companies like Solazyme in Arizona, where they're currently growing algae on 10-acre plots. And this application of my patent and my research would be able to make a large impact on the economic viability of their production of algae biodiesel and biofuels. Thank you all for taking the time to review my application.